So next up, we are going to have Sylvain De Guise. He is a professor in the Department of Pathobiology and Veterinary Science at the University of Connecticut. His research interests focus on the influences of man-made and natural toxins on the health of aquatic organisms. These include marine mammals, fish, lobsters, and oysters, and a specific focus on the immune system. So with that, I will pass it over to Sylvain.
Thanks, Danny. It's always hard to follow Ryan. First disclosure, and Ryan presented a breadth of work that has been done in the last decade or so. And I'm going to focus on a little bit more depth and looking into the mechanisms and trying to convince you that what we found was real and really associated with oil. My lab focuses on the immune system. Although we work with a lot of very smart people, dedicated people from a lot of different institutions to be able to collect those samples and to integrate the findings with the other aspects of health that have been discussed. So an overview of my talk today, I'm going to start with a little bit of background. Ryan provided very extensive background, so I'm not going to go in any depth in there. We're going to look into what has been previously described as part of the immune changes in dolphins following deepwater horizon oil spill. We're going to look a little bit more depth in the mechanisms that are involved and what does that mean? What are the implications for that? So like Ryan explained, we spent much of our summers in the last 10 years with 80 or so of my best friends doing live captures in bottlenose dolphins to assess their overall health with a team of veterinarians, technicians, scientists, and we spent a lot of time not only generating results in our respective labs, but getting together and trying to put the pieces together and understand what that means. So a lot of field work, but also a lot of work in conference rooms or in front of our computers to try to integrate those findings across our different labs. We sampled dolphins as part of this study in Barataria Bay that was within the footprint of the Deepwater Horizon oil spill. And we collected samples in the same time frame from a reference population in Sarasota Bay that is probably the best studied population of wild dolphins in the world. And they were not in the footprint of the Deepwater Horizon oil spill. And our lab, like I said, is focused on the immune system. And one of the very important functions of the immune system is the ability of your adaptive immune system. So the immune system that is specific, recognizes antigens, and then responds to those antigens. The primary response of those cells is to get activated and proliferate so that you don't have one cell, but you have a lot more cells to respond to that specific insult. And one of the ways we mimic that in the lab is we take blood cells, we put them in a culture, and we stimulate them with mitogens, CONI and PHA in this case, to stimulate the proliferation of those cells compared to unstimulated controls on the left. And what I'm going to present today is a series of graphs that look at Sarasota dolphins, the reference population, the first column in blue, and Barataria Bay dolphins sampled over time. So in 2011, a year after the spills, the first year we collected samples. And when we stimulate the cells of dolphins in Barataria Bay in 2011, they respond a whole lot more to the stimulus compared to dolphins in Sarasota Bay in blue. However, that increase in stimulation, in response to stimulation, appears to slowly fade off over time and return closer to normal in 2013 and 2014. So a change in the ability of cells to respond to a stimulus that appears to slowly return to normal over time. That was our first conclusion as part of that first paper. And when we compare it to the very few studies that look at experimental oil exposure, a study in mink found something that was very similar, that the response of T cells to a stimulation was increased over time after exposure to oil. So, and interestingly, about the same magnitude of increase in response. And a lot of people ask, is it bad if your immune system responds more? And as part of the National Toxicology Program, there's been a very extensive meta-analysis of tens of studies in lab animals. And the conclusion of that was that if you're responding more or less than anticipated, it's abnormal. And any abnormality in the response of the immune system can lead to changes that are not desirable. And a specific example of the ability of T cells of an increased response to T cells could have two consequences that could exhaust their response, and when they're stimulated again, they're less able to respond, or that could lead to in lab animals to other undesirable outcomes like autoimmunity. So when we continued to sample dolphins over time in 2016, 2017, and 2018, we find that we once again find an increase in the responsiveness of T cells to a mitogen, so that you stimulate them and they over-respond again. And that could mean one of two things. That could mean that dolphins got better and then got bad again, or it could mean that there was a steady increase in the responsiveness of T cells, but because we sample only a small portion of the population every time we do those live captures, that we might have missed the sustained increased responsiveness of T cells to a stimulation. But it's remarkable that it's so sustained in time and at about the same magnitude as we found in 2011. So we will at least call that a long-term disturbance of the immune system in those dolphins. And in order to verify that this was not associated with something else, we exposed mice to oil in the lab. And when we did that, obviously it's a much shorter time frame than we found in dolphins. But when we do reasonable exposure of mice in the lab, we found that there's not a real difference on the top between exposed mice in orange and control mice in blue. But interestingly, their offspring had a significantly increased response to stimulation of T cells in orange. So again, a mechanism of action that is consistent between dolphin sample in the field and very controlled experimental exposure in the lab, where there's nothing else that's different between exposed and control mice except exposure. So in trying to understand where does that come from, we looked at cytokine responses. Cytokines are small chemical messengers of the immune system. They're the means that cells communicate among each other, among different cell types, and sometimes stimulate themselves directly. And we find that there's some specificity to those cytokines. For example, on undifferentiated cells can either go on the left towards a Th1 response, and that is driven by a subset of cytokines. And that leads to a cytotoxic T cell response, so that the ability of T cells to destroy infected cells, for example, cells that are infected by intracellular pathogens like a virus. Or under different circumstances, different subsets of cytokines might lead on the right to a Th2 response. And that leads to the production of antibodies that will recognize and be effective at eliminating extracellular pathogens. So there's the other thing that's remarkable is that while they're complementary, they have a tendency to be a little bit selfish, and each response, in addition to enhancing itself, has the ability to inhibit the other response. For example, cytokines from a Th2 response directly inhibit the differentiated into a Th1 response, and as well, a Th1 response inhibits the further response of a Th2 response. And what we did is we measured those cytokines, those messengers that drive that differentiation in the blood of dolphins. And what we found is that the cytokines that drive a Th1 response in 2011 in barythera B were about half as low as in Sarasota Bay dolphins in, again, the control population. There were IL-12 was 20 times lower and interferon gamma was 4 times lower. So an overall inhibition in 2011 of the cytokines that drive a Th1 response, a cell-mediated response. On the other end, the cytokines that drive the differentiation towards the Th2 response were a little bit lower for the effector cytokines, but interestingly, IL-4, that is the cytokine that drives, the major driver of that differentiation of the direction of that differentiation, was 3 times higher in barythera B in 2011 compared to Sarasota dolphins, and those appear to resolve over time. So the big picture is that there's a complete downregulation of the Th1 response that is responsible for cell-mediated immune response, and there's a mixed response, but mostly an increase in the Th2 response. And when we look in later years, we find that there's a continued suppression of the cytokines that are responsible for a Th1 response, and there's a continued increase in IL-4 that drives the differentiation towards a Th2 response. So an imbalance in those cytokines. And there's another cell type, they're called regulatory T cells. And those cells serve at dampening the immune response, controlling, inhibiting an over-responsiveness that can be damaging. And in 2017 and 2018, again, just like in 2011, those cytokines in the blood of barythera B dolphins were much lower than in Sarasota. The other thing that we looked at, and Ryan hinted at that, is we looked at dolphins that were born after the spill. So they've never seen oil in their life, but their parents have seen oil. And we found that the dolphins that were born after the spill in barythera B had Th1 cytokines that were much lower than the dolphins that were born before the spill, or who were alive at the time of the spill. And that is a little bit puzzling because, like I said, those dolphins that were born after the spill have never seen oil in their life. However, they might have been, their mothers might have been pregnant or might have been affected subclinically by the oil at the time of the spill. And we did not find that large increase in IL-4 in those dolphins that were born after the spill compared to those that were alive at the time of the spill. The Treg, the regulatory T cell cytokine IL-10, was again much lower in dolphins that were born after the spill. We did further studies to develop and validate lab techniques to identify regulatory T cells by the expression of FOXP3 that you see in purple here that you don't see when we use isotype control antibodies. So we can quantify those rare cells in the blood of those dolphins. And interestingly, we found about 3 times as many regulatory T cells in the blood of barythera B dolphins than we found in Sarasota Bay dolphins. However, the cytokines that they produced were not 3 times higher. So that suggests that there's an overabundance of those cells, but they're not as functional. So the potential for a functional defect in those Treg cells. And again, very interestingly, when we expose mice to oil in the lab, and nothing is different, there's no other differences between the groups. The mice that were exposed to oil had over twice as many circulating Treg cells in their blood. However, there were no differences in the spleen. We went one step further, and when we looked at the not only what we find in the blood of those dolphins, but if we provide a physiological stimulus that promotes a differentiation of those naive T helper cells into either a Th1 response, or Th2 response, or a Treg response, do they have the same ability to respond if they're in dolphins from Barataria Bay compared to dolphins from Sarasota Bay? And what we found is when we stimulate the Th1 response, dolphins from Sarasota Bay, from Barataria, have about an equal response, maybe a little bit lower, but pretty much equal responsiveness to a stimulus to generate a Th1 response. However, when we stimulate a Th2 response, they're 2 to 3 times as responsive as Sarasota Bay dolphins. And that, again, fits the picture of a tilt towards a Th2 response in dolphins in Barataria Bay that would inhibit a Th1 response. And when we stimulate a Treg response, there's no difference between Barataria and Sarasota dolphins. And we push that one step further. And if we take Sarasota Bay dolphins T cells, we incubate them with physiological stimulus to generate a Th1 response. There's no statistically significant difference, maybe a very small increase with increasing concentrations of oil. But the Th2 response of Sarasota Bay dolphins that are exposed to oil in a culture medium demonstrates an increased responsiveness for a Th2 response, and there's no difference for Tregs. Again, if we put the pieces together, that suggests that oil, either in the lab or in real life in the Gulf of Mexico, oil exposure increases the responsiveness of T helper cells when there is a Th2 stimulation. So a pattern that is very consistent. So overall, what does that all mean? Barataria Bay dolphins exposed to deepwater horizon oil have modified T cell responsiveness, and the cytokines that direct the magnitude and the direction of those cells is also affected. It has a very consistent shift towards a Th2 response. And not only did we find that when we look at their concentrations in the blood of dolphins, but we also find that in lab experiments where we expose cells to oil. And those biases have been longstanding, almost a decade. The findings are also very consistent between dolphins that we sample in the wild and mice that are exposed in the lab. So that builds a weight of evidence that is very consistent and very supportive for the theory that oil is the responsible factor for those changes in the immune system. And what does that mean for those dolphins? Well, again, some meta-analyses performed by the National Toxicology Program in lab animals very clearly demonstrate that if your immune system is affected, you're going to be more susceptible to infectious diseases. So there's a very good chance that dolphins that swim in the Gulf of Mexico that encounter pathogens day in, day out, they're not always sick, but their likelihood of getting sick might be higher if their immune system is affected. And then the long-term aspect of those changes, including some aspects that appear to be more prominent in dolphins that were born after the spill, suggest that there might be continued exposure effects, continued exposure to oil in the Gulf of Mexico that drive those effects, or maybe that exposure stopped, but there's a potential for transgenerational effects, and that has been demonstrated with different contaminants in different species, including in our mouse study, changes in the proliferation of T cells that are found in the second generation that never saw oil. If that's the case, how long is that going to last? If there's effects that happen across generation or that are the result of continued exposure to oil, how long is it going to affect those? Is it going to affect those dolphins? And then what are the consequences for restoration strategies that might take into account the long-term recovery of dolphins? But if the second generation is affected, even if it's subtle, and those dolphins might still be young and might not have had the time to develop the long chronic effects that were found in their parents, how's that going to affect recovery over time? That's something that might be concerning. And I also would like to acknowledge the Gulf of Mexico Research Initiative that supported several of those studies, including the KERMA, which is probably the most stimulating consortium of scientists that I've worked with a very openness to collaborations and a diversity of results that Ryan presented earlier that are I think a hallmark of marine mammal studies when it comes to the effects of oil exposure. And with that, I will pause and take questions if there are any.